Hi guys, my name is Max and welcome to Simple Biology. In this video we're going to be talking about Hardy-Weinberg equilibrium. Now at this point you've probably noticed that biology is not very math intensive. For the most part this is because a lot of the things we teach in biology and a lot of things that we study are very hard to actually quantify. I mean, we talk about the ways that animals behave and some completely random seeming events that happen in genetics and in various other different studies that we have in biology. Hardy-Weinberg the Hardy-Weinberg equilibrium is one of the largest exceptions to this general trend. Hardy-Weinberg equilibrium is a way that we can actually quantify different proportions of an evolutionarily stable population, and you'll understand what that means later, but know that this is a great tool that we have in our study of biology, especially when we talk about evolution. So first off, what is equilibrium? Specifically, we're going to be talking about Hardy-Weinberg equilibrium, of course, but as you can probably guess, Hardy-Weinberg equilibrium is kind of a mouthful, so a lot of times we just call it equilibrium. And at equilibrium, the biggest thing that you need to understand is that the gene pool of a population doesn't change. It's completely static. Now, as you'd probably know, the gene pool is essentially the sum of all of the different alleles for a particular gene in a population. Now, of course, we're going to be talking about one gene at a time because it's really hard to quantify the entire genome of a population. So Hardy-Weinberg equilibrium is separate for each gene, and that'll be very important. The interesting thing about Hardy-Weinberg equilibrium is that when the population is in equilibrium, there's a very predictable proportion of the genotypes, and this can be observed using several equations that were actually discovered by these two biology people, whose last names were Hardy and Weinberg. That's where this equilibrium gets its name from. You don't need to know that, but that's pretty cool, I think. Anyway, these equations are what are used in Hardy-Weinberg equilibrium. Now, before I get into those equations, there are a couple conditions that we need to cover, and the reason for these conditions is that without them, no population can actually achieve full equilibrium. Another big thing, we will be dealing with various frequencies of alleles, and which are essentially different forms of a gene. Rather than the absolute numbers of these alleles, we'll be dealing with the frequencies. We'll actually define what these frequencies are and how we'll use them, but know that it's really just the proportion of those frequencies, of those alleles, to each other and to the group as a whole. So, of course, with evolution, it's only those proportions that will change. Now, here's these conditions. There are actually 5 of them that are generally accepted. The first and probably most important is that natural selection cannot be acting on the particular gene. This has obvious effects. If natural selection is occurring, then this will make it so that any unfavorable traits will start to decrease in the population, and that will change the proportions of the different alleles, of the different phenotypes to each other. And so obviously if natural selection is occurring on the population, specifically for this gene that we're looking at, then it will not be in equilibrium. The second is that there can't be gene flow. And so gene flow can occur if a part of the population moves away and goes to another population, or if another population sends some of its members over to the population we're looking at. And the reason this can occur is because this will just increase or decrease depending on what's happening, but it'll increase or decrease certain traits because no two populations are actually identical. Maybe you have one population of an island bird, and then you have another population of a bird that's living on the mainland. So obviously there are going to be some different traits depending on their environment, and if there's gene flow perhaps from the mainland to the island, those island birds will never actually reach equilibrium because genes will be constantly carried into that population, preventing them from reaching equilibrium. Our third condition is called preferential mating. And so usually this will occur with the female of an organism that will choose choose a male based on various characteristics. Maybe they're peacocks and they're choosing the male with the fanciest tail feathers, or if it's there's various other things. A lot of birds will preferentially select mates that looked like their fathers for various reasons that we're not really going to get into, but know that this exists. And what this can do is it will actually decrease the number of heterozygotes because they'll preferentially select mates that are similar to themselves or similar to family members. But most importantly, it'll decrease unfavorable traits for that preferential mating. So similar to natural selection, this sexual selection or preferential mating will have some sort of selective pressure against practically random traits, but it does have that pressure, and that will change that equilibrium. Next, no mutations can be occurring. Of course, this will randomly alter traits. Generally, we can almost ignore this one because mutations occur at such a minute scale, but if mutations are occurring, that will change the equilibrium in a way that we can't really predict. Next, there has to be an incredibly large population size. The larger the population size, the more likely it will be to be in equilibrium. And the reason for this is because there's a thing called genetic drift. And what genetic drift will do is over time, different alleles and different traits of a population can kind of randomly shift in one way or another. And with a very small population, it's more unstable and it's more likely to change at these random levels. Whereas with a large population, this genetic drift is less likely to occur. So again, if you have all 5 of these characteristics, all 5 of these conditions, then we can safely assume that the population is in Hardy-Weinberg equilibrium. So next, like I mentioned, we're going to define what frequencies are. And there are going to be 2 main frequencies, one of which can kind of be divided into 2. So you could say there's 3 main frequencies that we're going to be looking at with Hardy-Weinberg equilibrium. The first is the allele frequency, which I've mentioned earlier, but essentially the allele frequency is the total number of a particular allele divided by the total number of alleles in the population. So say we're going to start with this kind of very small population. Say you have a population of 2 organisms. One is homozygous dominant, one is homozygous recessive. You count up the recessive alleles, and that will be, there will be 2 because they'll both be in the homozygous recessive organism, divided by 4 in the total population would give you an allele frequency of 1/2. Obviously, we're going to be dealing with much larger populations than these, especially with Hardy-Weinberg equilibrium, but that kind of gives you an idea. Also, you can generally find the total number of alleles in a population by taking the population number and just multiplying by 2, because each gene will have 2 alleles. And then the next thing that we're going to deal with is the genotype frequency. Now, obviously, this is the frequency of a particular genotype divided by the whole population. So now we're just going to deal with the number of organisms in the population, and note that you have to know the genotype. So there are 3 genotypes, of course: homozygous dominant, homozygous recessive, and heterozygous. And so if we get the number of those organisms in a particular genotype, and we divide by the population number, we can find that frequency. Likewise, we have phenotype frequency, which is the same thing, but with a particular phenotype instead of a genotype. And the one thing we have to be careful with here is that if you have the dominant phenotype, that's actually 2 genotypes added together. That's all we're going to cover with frequencies. Moving on to the equations. These are really the only equations you'll ever use in biology, besides a couple others, but they're very helpful, and they're very simple. So I would strongly recommend you just kind of memorize this. P plus Q equals 1. This comes from these two variables, P and Q, represent the allele frequencies. Now, P we'll generally consider it to be the frequency of the dominant allele. For the purposes of this PowerPoint, I'm going to be using capital B, and Q is the frequency of the recessive allele. In this PowerPoint is going to be lowercase b. Now, P and Q don't necessarily have to be the dominant and recessive. They could be flip-flopped, but that's not really important, and if it's best to just keep it consistent. So I would recommend always using P for the dominant allele. Now this second equation is the one that's going to be more important for our purposes, and you get this equation by actually just squaring both sides of this equation. P plus Q times P plus Q is P squared plus 2PQ plus Q squared. And if you remember back from Punnett squareds, there's like a fancy Punnett square we can draw. If you have it weighted for particular allele frequencies, let's say that this is 0.3 and 0.7, 0.3, 0.7. Now these will be the same because you're making a Punnett square for the large scale population, and so the likelihood of getting one allele or the other, and I'm just choosing 0.3 and 0.7 at random. One will represent P and one will represent Q. But if these are the allele frequencies, then you can see that this is P squared, this is Q squared, and each of these is PQ. Because there's two of them, you'll have 2PQ. If this diagram doesn't really make sense to you, don't worry about it. It's just a graphical way of explaining it. Some people learn better that way, so I thought I'd share it. So next we're going to talk about the genotype frequencies, and these are where the bulk of what we're going to deal with is going to be talking about. Essentially, each term of this second equation represents one of the three genotype frequencies. First, we have P squared, which is the frequency of the homozygous dominant genotype, and this makes sense because it's the likelihood that they'll get the one dominant allele times the likelihood they'll get a second dominant allele. So P squared, which is the frequency of the homozygous dominant allele. 2PQ is the frequency of the heterozygous genotype. Again, you can refer back to that square, or it's the likelihood that they'll get a capital B and a lowercase b, plus the likelihood that they'll get a lowercase b and a capital B. You have to do both because there are two alleles, that's how it works. And Q squared works exactly like P squared. Basically, just know each of these three. P squared is the homozygous dominant, 2PQ is the heterozygous, and Q squared is the homozygous recessive. Now in our next video, we're going to be going over a couple different problems that will use Hardy-Weinberg equations. And so I would strongly recommend you go check out that video if you have any further questions, because that will be what applies the knowledge we learned in this video. But for now, we're just going to be dealing with it conceptually, and it's really just as simple as that.